Hello Bruins, and today I'm making a screencast on transcription and translation. So first of all, just quickly going over the central dogma of biology again. As you see that why is DNA such an important molecule in all living things, and that is because it contains not only the hereditary material that we say, but the instructions for proteins that run all living organisms. So here in this picture you see that the DNA is located in the nucleus, of course, in all eukaryotic organisms. And when you want to make a protein from DNA, you transcribe that information into a molecule called mRNA. And from that information the mRNA has transcribed, and then it goes out to the cytoplasm, to the ribosome. And there at the ribosome, the mRNA has the recipe to assemble the amino acids, which is, if you remember, the monomer of proteins, to assemble your polypeptide. Okay, and that's the classical central dogma of biology. Now, before we go into transcription and translation, let's talk about RNA. RNA and DNA have three key differences. One key difference is that sugar is different. So here, if you notice, between these two, this is ribose and has that oxygen, where this is deoxyribose. It does not have that oxygen. So this is found in DNA, and ribose is found in RNA. Second difference is the nitrogen base. In DNA you have thymine, so wherever you have A pairs up with T. A for adenine pairs up with thymine. But in RNA, A pairs up with uracil, and because it does not have thymine. And third, the shape is different. So in DNA, it has that double, we describe it as a double helix, where in RNA, it's still a helix, but it's like half a ladder. So we refer that to as single helix. So our three key RNAs that will play a major role in transcription translation is, first is mRNA. So this is mRNA, and here we represent these nitrogen bases, and each three nitrogen bases represents what we call a codon. Okay, and the second RNA is here in shape of like a hamburger, is our ribosomal RNA. And the reason why I show a ribosome, because ribosomes consist of proteins and rRNA. And rRNA is one exception I always talk about when I talk about enzyme. We also refer to it as a ribozyme because it has catalytic action. And a third RNA is tRNA, and tRNA stands for transfer RNA. And what it's transferring is, it has this complementary amino acid attached, and it matches up with the mRNA, the codon. So here we refer to as the anticodon. So for example, if our codon is AUG, then our anticodon is going to be UAC, and it will have the amino acid meth or methionine. So the DNA, remember, is always in the nucleus. So I always compare this to my grandmother's recipe book. So my grandmother's recipe book never leaves her kitchen. But if I want a recipe, I'd have to go take a piece of paper, write down the recipe, and that recipe that I write down is my mRNA, or I'm transcribing. So that process from DNA to RNA is transcription. And then once I got my recipe down, I'm going to go out to my kitchen and make my dish. And so here from mRNA has instruction, and here we are making protein at the ribosomes. So just a quick review on amino acids. Amino acids are characterized by R groups, and if you remember, there are 20 amino acids, and they are the monomers of a protein. And putting together these amino acids, so here's an amino acid, and if you remember, it has a carboxyl group and amino group, and there's a condensation reaction between the carboxyl and amino group, and when you remove that water, you form a peptide bond. So peptide bonds links amino acid to make a polypeptide, and that polypeptide will further fold and become a functional protein. So basically, DNA is transcribed to mRNA. So if you notice, these nitrogen sequence of nitrogen bases is a key to the genetic language. So every 3 bases is a word, and that 3 bases, which we call a codon, represents an amino acid. Okay, so remember, we have 4 bases and we have 20 amino acids. So if we only had 2 letters to our codon, that would have only made 16 words, so it would not have been enough. But with 4 bases, we have 64 possible combinations. So there's 64 codons, and these 64 codons represent what amino acid is. And if you notice, some of these codons have double or 4 for one amino acid, and if you notice, they kind of differ at the very last base. So if there's a mistake at the last base, it would not make a difference, or what we say is a silent mutation, and you would have that same amino acid. And some codons, one codon is a start codon, and then you have 3 codons here. That's a stop codon that tells you when to stop. Now, how do you use this codon chart? Again, this codon chart is based on mRNA. So here's your mRNA. It has a series of codons, and these codons represent words that translate into amino acid. So for example, AUG, so you look here, your first base A, so look here. Second, so look at the second base, and then you look down. So you're going to basically look at this quadrant, and then look for G. So G is MET, the start codon. U, here's U, second base U, third base U, and that's phenylalanine. So PHE for short. And then GCC, here's G, then look up here, C, and that's ALA for alanine. And CAU, so here's CAU, and that's his. Okay, so then basically at the ribosomes, you assemble these sequence of proteins. So this would be MET, phenylalanine, ALA, his, and this short segment of this polypeptide would represent a particular protein. Okay, so let's talk about first transcription. How does transcription happen? So first of all, you have a key player is DNA polymerase. So DNA polymerase will add RNA nucleotides. So remember how RNA nucleotides differ from DNA nucleotides. Got an I-ribose sugar instead of instead of T, it has a U, and it's single-stranded. So first, it'll untwist a segment of the DNA. So DNA will untwist, and then it will start transcribing by adding complementary RNA nucleotides, and it's growing in a 5 to 3 prime direction. So one strand of the DNA, this 3 to 5 prime strand, is your template DNA, and this other strand is a non-template. Okay, so basically there are 3 steps to transcription. The first step is initiation, where the RNA polymerase has to bind to a promoter, and the promoter is just a sequence of nucleotides where the RNA polymerase knows, okay, this is where the gene starts. So it will bind to the RNA polymerase and then start adding complementary nucleotides. And if you remember complementary nucleotides, wherever you'll have an A, a U will come in, and wherever there's a C, a G will come in, and so forth. And then when it hits a series of nucleotides, which we call a terminator, and a terminator sequence tells it this is the end of the gene, then everything, and the RNA polymerase will be released, and you have a completed mRNA. And again, this is a simplified version of transcription. Okay. Now in eukaryotes, you have further mRNA processing. What happens is you have these interrupting codons that don't code for a protein. So we call it intron. So remember, intron, in for interrupting. Then exons are codons that are going to be expressed, and they're going to become code for proteins. Then you also have the caps here in yellow. These basically protect the mRNA from enzymes when it's in the cytoplasm. So once these introns are spliced out, and then these exons are joined together, again, all done by enzymes, and then you have your final transcript of your mRNA, which will leave through the nuclear pores into the cytoplasm and to a ribosome. Also, there are key differences in transcription translation between prokaryotes and eukaryotes. And if you remember what a prokaryote is, so this is a prokaryote. Most prokaryotes are bacteria. They do not have a nucleus. So spatially, this DNA is not separated from the ribosome. So as soon as you go from DNA to mRNA, transcription, then translation cannot happen at the same time. So here's translation. So it's not spatially separated. Where in the eukaryotes, your DNA always resides in the nucleus, never leaves it, and then it makes this pre-RNA transcript, and then there's an extra step of RNA processing in eukaryotes, and then this final version of this mRNA will then finally go out of the nucleus to assemble those amino acids. So in eukaryotes, transcription and translation is spatially separated. And in prokaryotes, transcription and translation can happen. So as soon as transcription happens, translation begins. Okay. Now, translation occurs at the ribosome. So let's kind of go over key players